My channel Scientia WORLD. Hi everyone, now we are going to learn about osmosis. In detail, cell structure, osmosis, and there are two experiments on osmosis, that is potato osmometer, lab experiment on osmosis, which also explains the osmotic pressure. Okay, now comes to cell structure. Now we have to see the cell structures which are involved in osmosis. Okay, first one is cytoplasm, which maintains the concentration gradient for the movement of water molecules. Okay, next one is cell membrane, which is a selective permeable membrane and it surrounds the cytoplasm. And third one is cellulose cell wall, which is a dead material and it is permeable to almost all, that is for water and other substances it can be diffused directly without any selection. Okay, next one is permanent vacuole, which is a important one in osmosis, which plays a important role in osmosis. Okay, now comes to vacuole. This is the structure of vacuole and the membrane surrounds the vacuole is called tonoplast. Okay, and cell sap here indicated, which is also called vacuolar sap. Cell wall is freely permeable to water molecules and substances in solution. But cell membrane and vacuolar membrane are selective and they are responsible for movement of molecules in cell. What is the function of osmosis? That is movement of water. Okay, transport of water. So here water molecules we are taking as important, basic one. Okay, here cell wall is freely permeable to water molecules and substances in solution. Means if you kept any cell which have the cell wall, then the cell wall can diffuse easily freely the water molecules and other substances in solution. But the barrier is the cell membrane and again the vacuolar membrane, which are selectively permeable. Means they have some special proteins or protein channels, okay, which maintain the diffusion or concentration gradient. Okay, they are selectively permeable, means they involve the facilitated diffusion and they are responsible for movement of molecules in cell. Of course, because they are selective, they are responsible for the movement of molecules in cell. Next, vacuolar sap maintains the solute potential of the cell. This is the major function of the vacuolar sap. Okay, the whole concentration gradient is maintained by the vacuolar sap, which maintains the solute potential of the cell. Okay, based on that solute potential, the diffusion process may occur and the movement of water molecules also may occur. Okay, vacuolar membrane is called tonoplast, whereas cell membrane is called protoplast. This is about the cell structure. And what is osmosis? Osmosis is a term used to refer specifically to the diffusion of water across a differentially or semi-permeable membrane. Means osmosis is a term used to refer specifically to the diffusion of water. Means it is nothing but diffusion of water. Actually in diffusion we can see the concentration gradient where the solute particles can randomly move in the medium. But here the movement is only for water. It is a type of diffusion, but here the movement is for water molecules. Okay. In details, we can see the diagram here, before and after diagram of osmosis. If you see the before one, two chambers are there which are separated by a membrane. Okay, which allows only water molecules. The membrane couldn't allow the solid particles. Okay, if we observe the water level is same. Okay, after some time we can see in the after one that the water level changes. Okay, where the solid particles are more, the water transfer from the low solid particles having to higher solid particles, means from actually from lower water potential to higher water potential. But here something new is indicated, that is higher chemical potential to lower chemical potential. Okay, means chemical nature, which one have more chemical nature, means pure H2O. We have to know that our drinking water is not a pure water. It has some minerals and in it. The pure H2O is very bonding, having more bonding nature, having more chemical potential. Okay, so here the movement is from higher chemical potential, means having low bonds, means having low solid particles in it, to lower chemical potential. Okay, having more solute particles, having the bonding nature very low of water molecules. Okay, this is the process of osmosis. If we separated the two chambers by a membrane, the process is called osmosis, which is selectively only for water molecules. But in case if we avoid the membrane, the process is totally a diffusion process. Okay, this is the differences between osmosis and diffusion. Here come to the points. Driving force. As they involve the driving force, the water molecules. Pressure gradient and concentration gradient. This osmosis process is depends on pressure gradient and concentration gradient. We know that how the concentration gradient is happen, but how the pressure gradient is happen, we are going to learn in the second experiment. Okay, in detail. Passive absorption. Here the process is a passive process. There's no against the concentration gradient. Okay, plants absorb most of the water. Here we have to know that plants absorb water, we know, but most of the water is absorbed by this process, that is osmosis. Okay, and we have already seen the water movement, higher chemical potential to lower chemical potential. H2O having the more chemical energy and they are ready to bond. Yes, we have discussed about it, that pure H2O, pure water have more chemical energy and they are ready to bond. That's why they have higher chemical potential. Okay, equilibrium stage may happens. Okay, here maybe the equilibrium stage happens because it depends on water molecules, but if we see the in facilitated diffusion, there the equilibrium doesn't matter, because the diffusion is maintained by the needed of molecules. Okay, here equilibrium may happens. Osmosis can be explained by two experiments. Okay, now we have to come to the first experiment, that is potato osmometer. Experiment osmosis. Result movement of water molecules through membrane. Material potato, water, sugar solution, beaker and pin. Here the process is peel the potato having cavity, kept it in beaker having water below the height of potato and pin the potato above the sugar level. Then we can see the rising level of solution shows the net movement of water molecules. Means I will explain you it clearly. You can you can do it in your home also. First of all, we have to take a potato and peel the skin and having a cavity in it. Okay, and place the potato in a bowl or in a beaker having the water pure means drinking water as usually you are drinking, and kept it in a water of beaker, but it doesn't have to touch the level. Means there's no chance of diffusion. Means if you see the diagram before osmosis, the cavity, um, we have to see the cavity the water level couldn't rise the level of cavity. Okay, we have to care for it. And now pour the sugar solution, concentrated sugar solution, maybe 10% of sugar, into the cavity at some level, and we have to pin above the sugar level. Okay, after some time we have to observe that it crosses the level of pin. Means there is a rising level of water in it. It explains the osmosis, how it is happens. Okay, now we are going to the second experiment, osmosis experiment in lab. Result, osmosis, and also pressure affected the osmosis. Material, beaker, funnel, semi-permeable membrane, that is from egg, water, sugar solution, weight as a applying pressure. Okay, procedure, first we have to take beaker full of water, then insert the funnel having two sides open and tie a semi-permeable membrane to the bottom, and add at the bottom end and pour the sugar solution into the funnel at some level. Then we can observe the rising level of the sugar solution through the diffusion of the water molecules through the membrane is called osmosis. Okay, in detail I'm going to explain that we have to take the beaker. Okay, here we can see the beaker. Okay, having the water without solute particles. Okay, next we can see the funnel having the opening on both sides. At the bigger opening we have to tie the semi permeable membrane which is collected from egg. Tight it, then add a sugar solution in it into the funnel at some level. It normally we can see the rising level of water in the funnel. Okay, this is called osmosis. Okay, here the movement of water molecules may occur. This is the process which is involved in plants also. Okay, next we have to come to second one. Here the experiment is some different. Means there is external pressure applied on it, that is here 1 kg load which is taken as a pressure. Okay, if we apply against the solute particles, that pressure is called osmotic pressure. Here the pressure stops the tries to stop the osmosis, and that pressure is called osmotic pressure. Okay, osmosis when pressure applied. When external pressure applied on the sugar solution from the upper part of the funnel, there is a pressure built called osmotic pressure. No water diffuses into the funnel. What will happen if the pressure will applied? External pressure will applied on that. No water diffuses into the funnel. Okay. There is a barrier. The pressure creates a barrier, an external force which will stop the movement of water molecules into the funnel. Okay. Osmotic pressure creates. How it creates? By applying a weight on it against the osmosis, then the pressure creates, which is called osmotic pressure. This is the function of solute concentration. Because if we take the normal water, we cannot see the pressure. Means the water molecules easily diffuse into the beaker. Okay, but here the solute particles couldn't diffuse into the beaker because the semi-permeable membrane is not selective for the solute particles. That's why their role is to create the osmotic pressure. Okay, greater the solute concentration, greater will be the osmotic pressure. Means how much solute particles you are going to take against the osmosis, the pressure creates. Greater the solute concentration mean more the solute particles in it. They need more water molecules to maintain the equilibrium. That's why the pressure will be also more. Osmotic pressure is equal to osmotic potential, but in opposite direction. Means osmotic pressure is equal to minus osmotic potential. Just we have to know 2 points here. That is, semi-permeable membrane can get in an egg. How can it? We can get a semi-permeable membrane. Just we have to take a egg and having a small hole, which will remove which we have to remove the yolk and albumin part, and the remaining part we had to put it in dissolving in egg dissolving egg in HCl, means hydrochloric acid, for few hours. Then we can observe the outer shell of the egg is dissolving in it and leaving the membrane. Okay. Semi-permeable membrane can get in an egg. Remove the yolk and the albumin through a small hole at one end of the egg and place the shell in a dilute solution of hydrochloric acid for a few hours. The eggshell dissolves, leaving the membrane intact. Okay. In case sucrose solute particles does diffuse out through the membrane, will this equilibrium ever reached? Equilibrium may reaches, but through the diffusion and not from osmosis, because semi-permeable membrane doesn't act as a selective membranes to water molecules. Okay, that means if in case the semi-permeable membrane can diffuse solute particles also, then there is no role of semi-permeable membranes in osmosis. Maybe the equilibrium stage may reaches, but as per the diffusion, simple diffusion, that's it. It cannot play a role in osmosis. Means osmosis plays a role for the movement of water molecules. Okay, if there the movement of solute particles in it through the membrane, there is no a process of osmosis in it.